Now I invite Dr. Asatur Hovsepyan, Armenian Eye Care Project, Armenia, to give the e-poster presentation.
Hello, Doc. Hello. Thank you for invitation. I'm Asatur Hovsepyan from Armenia.
Okay. Please share your screen.
Yeah, just a minute, Yeah. This is my presentation. This is something from fundamental science. The name of my hypothesis presented it 3 years ago. It's named as particle wave dualism is questionable in many years. And this is my concept is a standing wave reception concept in the nature for color vision, also with photosynthesis and audio analysis.
Okay.
Today, the pigment theory of color perception is generally accepted, which is very superficially explains how the light acts on the protein molecules, opsins, named opsins, thereby giving preference to the particle theory of light. Pigment theory's color perception is generally accepted, but it is very superficially explains how the light acts on the protein molecules, named opsins, thereby giving preference to the particle theory of light. Although in the 17th and 19th centuries, the scientific world became more favorable to the wave theory of Huygens Young. This is Christian Huygens. You see the Thomas Young. Thomas Young was doctor too. And I'm speaking, my speak is very bad, but several days ago I lost my father. He's passed away. He's a good engineer and inventor from Armenia with Artsakh origin. And he always taught me in time of my education that I must go in the inside the processes, not explanations. Who, why, and saying why this process is going by my opinion. And so one time he asked me something was going wrong in physics. The light must have a wave nature, and the interaction with matter consisting of particles must occur only in portions of the light wave. It is the exposition of each portion of the wave with a short duration must be enough to influence each charged particle of matter and to force him to oscillate. He asked me, can I find that mistake in the scientific world? In time of my education, I told him that I found that mistake. This is a wrong mistake is here. Every educational book. So firstly, the scientists examined the effect of light on tissue preparations and not on living organs or vision. Secondly, the scaling of the task was zoomed into the level of proteins, molecules, and the science is very progressive to biochemistry or anything. But the wavelength of the visible spectrum is about 18 and 100 times greater than what is you see in this picture. So this is a market piece of light market as a light. This is not a light. This is a approximately X-ray shown. So all educational books must be changed, I think, because of my hypothesis. Based on the similarity of the anatomical microstructures of all receptor elements in nature, I declare that the light and sound must be considered as waves that enter the organism via special structures, refract within the internal media, and reflect back from special mirror, with final making of standing waves. These waves clearly distribute their energy at proper distances from the mirror, depending on the frequency of the wave. And the organism takes the energy just from their antinodes, where the oscillating electric field vector energy of the light or compression or refraction energy for sound is maximal. You can scan these QR codes. This is QR codes. My work is very big. It's about 40 pages. You can scan. First, it is going to my article, and second is to the two-dimensional animation I made for explanation. You can scan this QR code for more information later. I can't be in 20 minutes. Sorry. Let's go. This is a part of that animation I show you. Look at this is simple. For example, we start with the red light. Everybody knows that it's about 700 nanometers wavelength in the air, but it enters water or internal media of organism. It's changed the wavelength according to the refractive index, and making about 526. I calculated that everything. Finally, it go to the mirror, and everything about three colors. You see the all measurements for water and air. And the water is similar to the cytoplasm and every tissue liquids. So look at here the reflected waves making standing waves. You see standing waves with the different wavelengths. Finally, everybody can see the exact positions of the antinodes and nodes. You see, its names antinodes is the high energy points, and nodes is the zero energy points. So I made some calculations in my article. You can see that initiating electron, or for example, electron or oscillations. I take an electron that's a lower mass, and he must go and back on the oscillate in this region, and maximal speed it can't exceed the speed of light in the water. It's very big. You can't exceed. So all calculations I did according to that point. So finally, I established that all standing waves in the retina, for example, we are looking on the retina, the light coming to the membrane. The Bruch membrane is a very clear mirror, I think, and it reflects back all lights, and every light making his proper standing waves. You see here approximately 1, 2, 3 antinodes are clearly formatted nearby mirror, but after that, all energy distribution, some making some decaying because of mixed waves and interference. In my according my hypothesis, not only the shape of the, for example, L cones, M cones, and S cones is important. Also important the distance from that mirror. So look at here. For example, these red light antinodes now are very high energy here, and they must affect only the L cone. Other cones are not inflated. And the next, because for example, for green, only the green. On the blue one is for blue. This is the photoelectric effect showing that only green and or higher frequencies give only strongly perpendicular affecting of electrons going to the electric forces, electromotive forces. electromotive force is arising in the membranes of the disks, going to the surface direction. So electrons moving and oscillating here, making all photosynthetic or electric processes, biochemistry processes is possible. Also in the plantar chloroplasts too. Look at here, phylogenetically blue cones are very new, and they are located around the Maxwell's centroid, where are only red and green photoreceptors. So they included in the retina very strong directions, and this is making the polarization too possible because of electromotive force going horizontally to their axis, perpendicular to their axis. Everybody can consist that polarization plan must be here. So the blue cones are located in their positions only, or maybe by the open sides toward the center of uvula, or vice versa. It must be established histologically very carefully. This is a polarization plan making possible. The similarity of the chloroplast microstructure prompts that the light energy transformation is doing the same way. Reflected back from chloroplast internal wall, light created standing waves consumption free zones correspond to the green spectrum of the light palette. In addition, this concept explains why the color of leaves changed in autumn. While the contemporary botanics gives an inappropriate answer, that processes in the cell leading to the production of anthocyanin pigments. This is also pigment theory, but how the dying cell should spend energy and materials just to change the color of its clothes. This is a very look at here. This is showing the same in the plants. So they are thylakoids, making a complex named granum. Every granum has a distance from each other, and look at here. If why every leaf is green, because he passed the green light through him. This is the mean that about green spectrum doesn't affect photosynthetic process. And when the autumn go, the only dehydration process is making the color changes because of the loss of water, making a shrinking of the chloroplast, making him change color going to chromoplast, and green spectre consumptant too. And what changing palette of some blue and red lights, making amazing colors too. No, making membrane electromotive force the same way. It's perpendicular to the light propagation vector. Interestingly, the nature goes the same with the perception of the sound. The standing wave concept explains how the sound wave passes through the internal air liquid perilymph, then reflects back from a round window and creates a standing waves palette. Note that sound speed is diminished in these air compartments due to jelly proteins, and I can't find any data about mechanical properties of the perilymph. It's only biochemical parameters are available in the science reports. So look at here, this is jelly, just you see the coming inside waves in the jelly. They're very lowering the sound speed. So next, this is my father's and me audiology Soviet patent, naming that Asatur certificate. This is the receiver of sound signals. Honestly, we did this work is very in 1992. It's old work. It used the standing wave principle too. This is a analyzer for sound. Each microphone is established for each frequency. It's for sound. Look at the last animation. Sorry, the sound going to the outside, entering the air. The sound is not longitudinal. Is a longitudinal wave. Sorry, because is has a contain moments of rarefaction and compressions. So that's just schematically we explain the sinusoidal wave here. So look at here, the sound wave entering the membrane, is oscillating the tympanies, and through the mechanism of small osseous structures, giving that the same oscillation to the oval window membrane and entering the perilymph scala vestibuli, going around the helicotrema and going back to the round window and then reflected back. So this is a showed section view of the cochlea. And when we open the cochlea, just with both scala, vestibule, and tympani, and show the frequency distribution here, you see that this is an interesting thing. The pressure is making several diapasons available for our ear. The longest wave antinode is possible to hear by human is 20 Hz. The middle part of the which is proved by histologists is the zone of the helicotrema. It's about 200 Hz, and the maximum possible hearing voice is 20,000 Hz. It's very near, close to the round window. So making calculations, you see in my article, you can see in my article, the oscillating of some antinode, giving a movement of the both membranes. Basilar membrane, it depends on the location of that antinode. So if the antinode is about between 20 Hz and 200 Hz, is a low frequency voice. It's located in the scala vestibuli, and pressure giving the oscillations of Reisner's membrane. So small calculations give me that about, for example, 1.1 centimeter distance from the round window, giving us to hear 305 Hertz sound. So look at here in cross section, it's possible to hear the amplitude of the voice. The amplitude is detected by involving each neighbor hair cell, which is located in the same distance there are four, about the four or three heart cells in the full organ corti. Look at here. The firstly, with small sound, starting oscillating the first far heart cell, and if the sound become more loud, the second heart cell involved too. If you give a more louded sound, the third neuron heart cells they also oscillating and making narrow signals to the brain. Look at here. This is a maximal amplitude of oscillations in scala tympani, giving such picture. This is for frequencies between 20,000 hertz to the 200 hertz. If the membrane of Reisner's moving oscillations, it's another sequence going to the brain. For example, the same neuron, the same heart cell, if fourth heart cell is oscillating at first, it demands that about 105 hertz sound is hearing, acting. And if the membrane of Reisner's hitting more loudly, it's the third neuron coming in function. And if the membrane of Reisner's is going to the more louder, the opposite direction of sequence is going. So finally, the spiral ganglion going to the center of hearing. I placed the three in the same picture. Look at here, the frequency of red light here is hearable, is detectable for red cones. It depends on the distance of the between membrane bruch of and center of photoreceptors, and the same in the plantar thylakoids. Look here, and the same is for sound waves. Sorry, I'm finished. Thank you.